So hi, hello everyone again. Hi, hello, Microbehunter here. Welcome again to another Saturday live stream, a Saturday microscopy live stream. Well, so I hope that as always, all of you are able to properly hear me. And I had some echo. Okay, here we go again. Yeah, so here we are again. Today I'd like to answer again a few questions that are collected over the last couple of weeks, microscopy questions. And I see that some of you have already started to type in the chat. And as always, for the first couple of minutes, let's hi all across the world. A hello from Lebanon, from Costa Rica, from Vietnam, from the United States, from the UK, from Switzerland, from Scotland, from the Netherlands, again from the Netherlands, from France. And somebody already at the beginning here, Teeny, wants to see a couple of water samples. Well, actually today I would like to talk mostly, about some of the questions here. But I mean, here is a little rotifer that I just put under the microscope right now. So, and while I'll be answering some of your questions, some of which I already prepared and some of which you might be asking, I'm of course going to show you a little bit, some of the slides that I have here as well. Okay, from Paris, from Turkey, from Berlin, yay. Okay, yes. By the way, you already might know this little critter here. That it's of course a rotifer, and and it's more or less happily living along and eating some food. So over here, this here is my desktop view, and look, I've prepared a whole bunch of questions here. Where did I get the questions from? Many of the questions were posted in my YouTube videos. I already responded to some of them. Sometimes I got some emails with a few questions, and I simply kind of collected them. There are a lot of them. I don't even know if I have time to cover all of those. But in any case, I will try to go through those. Of course, I will interrupt myself when you have questions and when we're posting some questions in the chat. Then I would also, of course, like to talk about those. Okay, hello from Australia. Have you used super glue to mount slides? No, I have not used super glue yet. I think super glue requires water to solidify. Okay, so this is something that it might take therefore a longer time, because if you put it between a cover glass and a microscope slide, then I think this might cause the thing not enough water to reach it, because super glue glues because solidifies because of moisture. Okay, and hello from Texas. So pretty much randomly I would like to start here. I mean, many of the questions that I received here are from I don't know, about microscope buying questions and so on, and I'm kind of randomly choosing now a question here. This one already relates a little bit to water samples, so I'll also be talking a little bit about some theory maybe, and we'll see how where the live stream is going to take us. Okay, so this poster here, viewer here, asked, I'm interested in seeing what kind of bacteria live in my aquariums. Are they the same? Is it drastically different between each tank? What's it like compared to natural water? That's what I want to have a microscope for. Well, of course, always a big thank you. Aquarium microscopy is indeed something that I also recommend. However, I think bacteria are not among the most interesting specimens here. But I think I can indeed answer the question, are the bacteria the same? Well, some bacteria certainly will be the same. Of course, we were talking here about an aquarium here, but it is unlikely that the conditions in all aquariums are exactly the same. And therefore, what we're going to see is that there will be different bacteria in the different parts of the aquarium. In the water, there will be different bacteria than, for example, in the sediment, than, for example, in the filter. So there are, of course, different parts of the aquarium will have different bacteria. And another important thing here, and I think that's the most interesting thing, is well, actually, I think a microscope is not very useful to distinguish between different types of bacteria. It might be a little bit surprising to you, but the shape of bacteria only says not a lot about which type of bacteria they are. The biodiversity of bacteria is incredibly large, and just because two bacteria look the same does not mean that they actually are the same. In order to really determine whether bacteria are similar or different, you have to do a DNA analysis or you have to do a biochemical check. Yeah, so if this is the reason why you would like to get yourself a microscope, then I have to tell you there are many, many more interesting things to observe in an aquarium than bacteria. Okay, but basically the answer is an in between. Some bacteria will be the same, some will be different, and I think that's maybe the most interesting part, most bacteria that occur in nature have not even been identified yet. So there will be certainly a lot of bacteria in your aquariums that have not been described yet. Okay, so yeah, hello from Canada. Is there a budget model trinocular microscope you might recommend for beginners? Okay, hello from Massachusetts, from the West Coast of the United States. Okay, from Argyle. So yeah, is there a budget model trinocular microscope that I might recommend for beginners? Well, there is no specific model that so I'm fine. There is no specific model, single model that I recommend. I would say, I mean, I've in my basically on my Amazon web affiliate shop, there are a few models that I collected, and they cost around $300 to $400 or euros. Whether you're satisfied with those or not depends a lot of course, also on your personal preference and expectations. Okay, so generally a trinocular model that yeah, you have to count approximately $300 to $400 or euros and above. Okay, yeah. I believe microscopes are being used to identify koi and aquarium fish diseases. Well, there are, of course, certain fungi, certain skin infections of certain fish, and if you are able to isolate certain fungi, then of course you can try to make a diagnosis. But then you're already looking for something specific, like for example for certain fish diseases. But if you want to make a general comparison about bacteria that exist, then I think that's going to be a little bit more difficult. Okay. Thanks for the video. So this was a comment of one of the videos, and you know what, just to keep life a little bit interesting then, or the stream a little bit interesting, I will be switching over then also again to the microscope. How about the stream about the detection of microscopes using a microscope with UV light or other technologies? Microplastics. So I misread. Using microscopes with UV light. This is actually something, the reason why I chose this question is because I actually wanted to do something like this already before. Microplastics are an issue in the environment. So for example, many synthetic fibers from clothing over the washing machine, they end up in the water, and those microplastics can be identified. And indeed, I was actually thinking about maybe starting some kind of a worldwide citizen science project where people all over the world try to collect microplastics and document it with the microscope. And you can do that by actually staining the water sample with, I think it's called, the dye is called Nile red, and then it will start to shine in UV light. But not everyone has a microscope like this. So what I think about doing is I think about maybe making a method which allows people to use an ultraviolet flashlight to detect microplastics. I'm still working on this, but I think it's a very interesting suggestion, and maybe if some of you have already worked into this direction, please share your experiences with that. But microplastic detection in water samples and so on, I think is really an interesting thing that can be done. Okay, so but yeah, let's quickly go over again. Here, that's again the water sample from before. You can see. Yeah, look, there are lots of single-celled microorganisms around. It seems to be like, oh, what is this here? Very interesting, yeah. So this water sample here was already a little bit older, and over the time you can actually see that certain microorganisms start to appear and others start to disappear. And you see, just by looking at the shape of those microorganisms, these are not bacteria, these are too large for bacteria, but just by looking at the shape, you cannot really say always if they are what they are. Yeah? You'd have to, yeah, sometimes you can, but sometimes the information that you receive over a microscope is not enough. Okay. So hello from Barcelona as well. So what else do I have here? Yeah, this is something This is another interesting question here that keeps on reappearing a little bit. Yeah. Is it possible for you to do a comparison between Chinese-made objectives, microscope objectives, and those made by a brand, for example, Olympus? Yeah, the point is the following. It's an interesting question again, and because in the microscopy community there's this term called a China microscope. It's because microscopes are made in China, and sometimes this is used to refer to low quality microscopes. Well, I gotta be honest with you, these days pretty much everything's manufactured in China, and this is really not a quality criterion. I mean, of course, microscopes manufactured in China, they can range from extremely high quality to very low quality. Everything is there, and it really depends very much on the brand and on the importer who imports the microscope on the quality. And basically what I've got over here is this here is a most likely made in China, but it's an objective that's very common, and it's a very economical, very low cost objective. And but still they will produce generally fairly good results. Okay, and if I now compare a good objective with a bad one, then of course it could be that if I get a very old Olympus objective from the 1970s or 80s, then it could be that modern China-made objectives are actually much better. Yeah. So you see, it depends really on the individual objective and also on the price of the objective, and it's very difficult, if not impossible, to simply compare this directly. You have to look at the individual case. Top-down and cross-section of sample comparisons with the microscope might be interesting. It will require more thorough involvement. I see. Basically, to have a sample cut in different directions. That's indeed a possibility. I might have some slides here where I can show it to you, but just for the fun of it, I'm just going to move over here. In case you're wondering what this is here, this is a cross section of maize. Maize is corn, and this is the vascular tissue. You can see the individual cells here. So this is where water is transported up the stem. I like the plant cross sections a lot because they look very beautiful. So there is a recommendation. The Swift SW350T. Okay, it's good. So there is the 350 and the 380T. The T stands for trinocular, so this refers back to one of the questions of one of the people who commented about an introductory microscope, a trinocular one. Okay. Hi from Switzerland. Are there even objectives with a 15 millimeter diameter? Are you referring now to the? Are you referring now to this? To this here? Well, most of the objectives have also called an RMS threading, which is, to my knowledge, 19 millimeters. There are smaller ones as well. I don't know if they're 15 really, but the introductory microscopes or children's microscopes, they have actually a smaller standard where the diameter is smaller. Yeah, but most, at least most microscopes with 160 millimeter objective optical mechanical tubular length have the standard threading, but there are smaller objectives also available. Yeah. My Amscope stuff from China is quite nice, even have phase contrast. As I'm saying, it depends a lot on the individual case, and these days many things are manufactured in China. I remember once we ordered for school several years ago Leica microscopes, the German brand. Okay, because we wanted to have good brand microscopes, and they were good, no question, right? But it was kind of fun. We had quite a big laugh because on the box and on the microscope there was a "Made in China" written on. Yeah, so we bought a German quality made in China. We had a pretty, we biologists in school who, when we unpacked the microscopes, we had a good laugh. Yeah. Hi Oliver, is phase contrast device very much better than phase sliders? I'm a little bit confused by that question here. I think you're referring to maybe a slider in the condenser, and maybe one where you can rotate the condenser. I'm not quite sure what you actually mean with that. So I'd have to ask you please to be a little bit more specific here. What microscope are you using? I'm using an Olympus microscope. My first Olympus I got 25 years ago, an Olympus CH40. which I'm still using. It's a very nice model. I've got then also Olympus CHA microscope, a used one from the 1980s that I like to use. So I've got several ones, and I keep on changing them. What microtome do you use to make the thin cuts of the plants? Well, I do have a couple of microtomes as well, also homemade microtomes. I'm not always happy with the microtomes that I have, but what you need to get is you need to get yourself a very sharp knife. But you know what? If you wait 10 seconds and I'm going to get you the microtome to show it to you. So here I am again. Okay. Yeah, because you were asking, this is the one that I'm using, but yeah, the quality depends quite a bit on the sharpness of the knife. Some of you might have already seen this before. The box is not so nice looking. I always have to, honestly, I have to laugh so much. I had to laugh so much when I opened this knife here, which is supposed to be very sharp. It is sharp, but look at this. If you open it, what a disaster. It's totally useless. Yeah, it's not even straight, and there's this black dot here. Yeah, it's yeah, it has to be properly sharpened. This is really yeah. I kept it simply as a bad example, but the microtome itself is quite okay. Yeah, it was a fairly low cost one, and it does work. And but what you need is you get yourself a very sharp razor blade. I'm using this one over here because it has a built-in clamp. I've got another microtome which does not have a clamp, and this makes it a little bit more difficult to use. I've also made some videos in the channel where I experimented by actually making my own microtome, which also works. I would say the success of a microtome depends also quite a bit on how many tries you make, so that you're going to be successful. Okay. Technically, majority of them are made in China, including the big four. The knockoff version directly from China will be a lot cheaper. The quality could be very poor in general and might not last long. Yes, so to clarify, is the following. Is the big four. That's a term that, I've also been using a lot. These are the big four companies, Olympus, Zeiss, Leica, Nikon, the traditional big four manufacturers that make microscopes for research and science and so on. And even they manufacture some of the parts in China. But the quality, of course, is great. But again, it depends very much in the individual case. Out of curiosity, can you distinguish an animal cell from a plant cell under the microscope? In general, yes, in most cases. It depends if the cross section actually has all of the characteristics that you actually want to look at. These here, for example, are plants, and there's one thing that is very notable, and that's why you're able to distinguish them, and that is, now I've got the arrow, you're able to see that the thick cell wall. Okay. So, and that is something very, very easily visible in plant cells. If you now have an animal tissue under the microscope, then because there is no thick cell wall around them, sometimes the cells are sticking together so closely that it's difficult to distinguish the individual cells. Okay, and there are also other characteristics of plant cells, and it depends if you are actually able to see them. For example, the presence of chloroplasts when they're green and so on. But generally, this is one of the reasons why the early microscopists, they found out quite quickly that plants are made of cells, because you could see the individual cells easily because of their thick cell walls. Yeah, but for example, if you look at the rotifer from before, let's see if I'm able to find it again. Okay, let me refocus here. Maybe if we're lucky, we're able to find again one of those rotifers. Where are they? They are actually micro animals made of multiple cells. I read somewhere around 1,000 cells. But seeing the individual cells is much more difficult. See, now they're gone. Here's one. Here two. Okay. Here three. Okay. So those rotifers are micro animals. Okay, and they are made of multiple cells. And here, it's not so easy to actually see the number of cells. As a matter of fact, adult rotifers of a certain species even have the same number of cells. So that basically, it's an illustration that here it's much more difficult to see, if not impossible to see the individual cells. Okay, so let me see. Can you make your own microtome? Has a video about that, and I think a whole live stream about microtomes too. Yeah, I did experiment around with some microtomes. I 3D printed one as well. Yeah, it's not so difficult. You use a screw. You can buy stuff like this in a hardware shop, and that's not so difficult. Hello from the UK. Thank you. Yeah, I just got a new microscope with phase sliders to insert the condenser. That's cheaper than with the micro with phase contrast. Well, phase sliders. Well, what you need is you need, I mean, I guess the phase sliders or the phase annuli, which go into the condenser, and you need that, and you need phase contrast objectives. Yeah. Yeah, £2,000. Yeah, I bought a 60 times objective, but I can't move the stage up far enough for the sample to come into focus. It crashes into the lens before the sample comes into focus. It's fine with other objectives. Any ideas? First of all, I will tell you the following, that I've got a similar case over here with my microscope. I just want to show you. This is now 40 times. Now look what happens when I rotate the 60 times objective into place. Yeah, and sometimes, now maybe not here, but sometimes when I move and when I focus, you don't see it here so much. Then, as a matter of fact, the lens of the microscope of the objective will actually also push against the cover glass. So this can happen, especially if the water film is a little bit thicker. However, if it really always crashes and if the distance is always wrong, then it could be that the objective, the 60 times objective, is not of the same series as your other objectives. So parfocality is not there. Normally you should have all objectives from the same series so that you can change the magnification between them and you do not need to refocus. But it could be that this 60 times objective, if it is from a different series, has a different so-called parfocal distance, and therefore it might bump into the slide. Okay. Hello from Ontario. Do you think what the Pressa Infinity 301 is a good microscope? Look, these questions I get a lot. Okay, is a certain microscope a good microscope or not? As a matter of fact, I get them, I don't know, several times a day. And I gotta tell you, whether a microscope is a good microscope or not does not only depend on the microscope. It sounds kind of weird when I say that, I know. Whether a microscope is of sufficiently high quality or not also depends on you. Okay, I just wanted to give you an example of some of the microscopes that I even put into my Amazon affiliate shop. Some people said, "Wow, such a good microscope for only $300 or euros. Wow, isn't this unbelievable?" And then other people say, "Oh, come on, for $300, that's all you get." I mean, it's just they find problems everywhere. So the truth is somewhere in between, and it depends a little bit on your own expectations of what you want. Now, this answer does not satisfy you, so I'm going to give you a slightly better answer. Generally, microscopes that have infinity optics are more expensive, and usually are overall of a good quality. This does not mean that microscopes who are not infinity optics are all not of a good quality, okay? But simply because they are more expensive, sometimes also targeted towards the research and medical field. Therefore, usually infinity microscopes are also otherwise, not only optically, but also mechanically quite okay. Yeah. What is the best way to clean eyepieces? I don't have it here. Do I have some? Yeah, I don't. The best way to clean eyepieces is as follows. Don't clean them unless necessary. Okay. Yeah, I'm just gonna use this here as an example. I don't have the solvent here right now with me, but see. No. I have to get again. What you do is just a second. Let me try to find some cotton swabs. So for cleaning eyepieces, what you need is the following. Okay, those cotton swabs. And you need some alcohol, isopropyl alcohol or simply ethyl alcohol works. And what you do is you only do that to carefully clean away the grease and the grime from your eye and eyelashes here. Okay, and what you never do, you never use any commercial cleaning solvents like cleaning eyeglasses or windshields or windows or so. This is a no-go because they are not sufficiently volatile. You need something that evaporates quickly. So you take some pure alcohol, and what you do is you do not put the alcohol on the eyepiece. Okay, because otherwise it's going to run in, right? You put a small drop of alcohol over here on the cotton swab. You make sure that it's not dripping. And then what you do is using a rotating motion, you carefully, clean off the grime. You don't use lens paper. The reason is because lens paper does not have a very high surface area, and therefore if there's some I don't know, dust grains in here, then you might end up rubbing it into the lens. But the high surface area of the cotton swabs allows you to pick up any dust and dirt. Okay, and you do that, and it'll be fine. Okay. So rule number 1, large surface area of cotton. Rule number 2, no solvent, no alcohol on the eyepieces itself. And rule number 3, you do not use any commercial cleaning agents that are made for windows, for car windows, for monitors, for eyeglasses or anything like that. It's a no-go. Okay. So wow, I'm getting so many questions from the live chat, which is fine. Okay, but I've got some more questions here as well, but I'm going to give the live chat questions a priority. Do you have any experience with Motic microscopes? Yes, I do. As a matter of fact, same here. Motic is a company that is apparently the mother company of Swift. Okay, at least sometimes they have the same webpage, the same address. I also made in this channel, I also made some years ago, put a Motic microscope and a Euromex microscope. I unpacked both of them. Okay, so I made a review of some of them. So you have to check in this channel in the unpacking and reviews playlist. You're going to find also a review of a Motic microscope. But again, I mean, it depends on the type of model that you have. Yeah? What is the plasmid ring-like structure in your plant sample? The plasmid ring-like structure. I have to tell you, I'm a little confused now. Okay, that's again the thing here. I didn't even get to change over the... You mean, I don't know, is this the thing that you're kind of referring to here? This is the so-called the vascular bundle, and those cells here, that's the xylem. This is where water is transported up the plant. I think this one over here must be the phloem, where substances are transported down the plant. Okay, if this is what you're referring to. Okay, are onion cells alive? Well, yeah. I mean, unless you prepare them to make a permanent slide. Onion cells are of course alive, just like all living things are alive. I mean, all plants are alive. Okay, so yeah, sure. If you put up onion cells, for example, under the microscope, then you're going to also see movement. Yeah. You should look at the Microbehunter's live stream about phase contrast microscopy. It might help you. Yeah, I did one last week about phase contrast. Yeah. Oftentimes 60 times objectives are spring-loaded, depends on the objective, and some are. I just want to show for those of you who don't know what spring-loading is, here this is a so-called spring-loaded objective, and see that the front part can be pushed in so that if it crashes into the slide, okay, then the front part retracts. And this kind of protects the front lens. However, I also have to say that in many objectives, the front lens is a little bit deeper in any way. So you will, even if you crash into the slide, the glass surface of the lens will not make contact, but rather the surrounding metal here will make contact first, because the central lens is always a little bit either concave or a little bit set back, so for protection. Okay. I also bought a 60 times objective, but I had to lower the stage. Yeah, that's the case when the objective is not parfocal with the other objectives. Okay. Hello, can you buy a different brand objective than it comes with if you change all of the objectives? So basically, I see. So basically the question is you want to exchange the objectives of your microscope, and are you able to do that? Well, if your microscope, you got to be careful here. If the objectives, first of all, can you mix objectives? Yes, I have also done this. I have also bought myself a 60 times objective once, and the distance was also wrong. But I knew that, so I always had to remember how many times I had to turn the focus knob until it got into focus. So I also had to lower the stage. So that's not a problem. I was experienced. I could do that, right? Now, what happens if you want to completely upgrade all of your objectives for whatever reason? Are you able to do that? Well, if your microscopes have a 160 millimeter standard, then generally you can do that. Okay, unless you have very short objectives. There's certain introductory microscopes that have so-called short barrel objectives, which are even smaller. Okay, but yeah, because the 160 millimeter standard is generally compatible to each other. Okay, you cannot simply upgrade if you have so-called infinity microscopes, because they might physically not fit. And there's also an additional optics, the so-called the tube lens, which must be compatible. But generally what I would do is if you've got microscopes with infinity, then I recommend that you contact the manufacturer, because they should be able to provide you with spare objectives. Okay, so I hope that this kind of answered it. Yeah. So in other words, I have done this already. I bought myself a whole range of new objectives and I simply exchanged all of them. It's not a problem. Hello from Kansas. I just ordered a microscope. I got scrolled. Ordered a microscope and can't wait to get it. Just found this channel last week and you have been such a help. Well, happy for that. For those of you who are new, I'm doing weekly live streams here, some product reviews here, but if you're also interested a little bit in simply observation, then I do have another channel as well, simply called Microbehunter. I've just today reached 90,000 subscribers on that other channel, which you also might want to visit. I've been experimenting with YouTube Shorts, and I've been also simply doing a little bit of nature observation. It's less technical than this channel over here. Okay. I just spent 4,200 Canadian dollars on a microscope and a 5 megapixel camera. Yep, that's a nice one. Okay, hello. You did a video using capsaicin to inhibit alternate movement of paramecia, yes. Was it specifically a hot pepper, or does it even mild yellow or green bell peppers work as well? I think I used a hot pepper for that. Honestly, I don't know, I didn't try others. Yeah, specifically, if you really want to make sure, what I've done is, for those of you who have not seen that video, I put some paramecia, some single-celled microbes under the microscope, and I added some of the juice of a hot pepper, and then the microbes, they didn't like that. They kind of died. Specifically, you have to actually get the pure substance and test it out, because it could maybe be a different substance inside the hot peppers that caused them to die. Okay. Yeah, I ordered last week the Motic BA310E trinocular with the Moticam A5. Yep. Is Baader Optical Wonderful Telescope okay for lenses and eyepieces? Again, I don't know Baader Optical, and I would again say very often many companies will sell microscopes and telescopes with a wide range of different quality and price. Okay, so again, here I would say is you have to look at the individual brand. Now, another suggestion that I would give is the following. Maybe this kind of also, how do you say, influences the type of brand that you want to choose. It's not necessarily only the microscope, but also the support that you get. Okay, so for example, if you want to upgrade the microscope, do they offer objectives? Do they offer phase contrast upgrade if this is something that you want? Okay, replaceable eyepieces or whatever. Okay, can you exchange the LEDs or the lamp? So just wanted to tell you that just recently we're now servicing some old halogen microscopes in our school, and yeah, one of the teachers, not me, but one of the teachers wanted to replace some old light bulbs, and he said, "This is the old light bulbs. They're so difficult to get, so he had to order some from outside of the country." So all of the service and this whole thing might also be important. Okay. Correct. On the left side of the structure that you pointed out, if you zoom further in, you will be able to see individual plasmid ring-like structures. Honestly, now I also want to know, on the left side, I don't know, do you mean these here? Okay. Yeah. Well, all of these structures that you see over here, these are individual cells, okay? The cell walls, and these are the so-called the vascular bundles. Oh, you mean those here, those ring-like structures. Yeah, I wonder if these could be not the reinforcement rings of the xylem. Okay, a xylem is are cells that transport water up a plant. And what they often have, and I've also made videos, is sometimes they have a spiral-shaped reinforcements or ring-like structures in them, which kind of make sure that because of the water pressure, the cells don't collapse. And if you cut it across, then it might appear to be like this. Okay, so I suppose that these might be those ring-like reinforcement structures of the xylem. Yeah. Yeah. I just skip over some of the questions here. 5 megapixels is perfect. Hello, just saying thank you for creating this channel. I'm happy if it's useful. I'm an engineer in a French startup. Thanks for your videos. We bought an Amscope microscope and it works perfectly. Okay, well, that's nice. For microscopy purchase, it varies with the individual. It depends on the type of applications, of the cost, the upgradability, on the objectives, other instruments per future potential application. And I would like to add another thing. The quality of the microscope also depends a lot on, yes, it sounds weird, emotions, personal preference as well. Okay, yeah, there is certainly also a certain emotional attachment, and sometimes you simply like to use certain microscopes more than others. Especially if you're doing this as a hobby, this can be important. Okay, so no lens, so that goes back to the eyepiece cleaning. No lens tissue and isopropanol on used oil lens for quick cleaning. Hard to see where Q-tip is going upside down, and does isopropanol affect lens coatings over time? So the thing is the following. Could alcohol affect lens coatings? Possibly, depends on the coatings, but generally alcohol, or at least ethyl alcohol, has been recommended for cleaning optical surfaces. And the reason is that it is not too aggressive. You should not use any other solvents like xylene, which can be found in some paint and so on, because they are really able to damage it. But generally, what I read is that fast evaporating alcohol should not be a problem. Okay, and normally the lenses should be designed in such a way. Yeah? When buying a 60x objective, it is important to check the working distance. Some objectives have such a short working distance that becomes almost useless only for commercial slides. So basically what this means is the following. The working distance refers to the distance between the objective and the slide. Let's just use a slide here. Okay, so this is basically, this is the working distance, and depending on how thick the specimen is, for some 60x objectives, you might actually crash into it, because the distance is so small. Yeah. So this is indeed an issue, and if you use 100x oil immersion objective, then the distance is even smaller. This is actually one of the reasons why I say that in many cases a 60x objective is actually in many cases more useful than, let's say, 100x oil immersion. Yeah. Do you know if a MP250 pocket microscope? I don't know it. Sorry. I don't know this pocket microscope. Okay, how do I tell if the proper working distance is? How do I tell what the proper working distance is for my scope? Well, quite easy. The working distance is different for each objective, because that is the distance between the slide and the objective. I guess what you're talking about is the so-called the so-called, how do you call this, the not the parfocal distance, it's called the conjugate distance. That's how it's called. Okay, the conjugate distance is for most microscopes 195 millimeters, but that's a technical term, which should actually means that all of the microscope objectives have a conjugate distance of almost all of them of these of 195 millimeters. These are basically the standard size ones. So I would say the working distance should be irrelevant for you, because if the microscope is of the same, the objectives of the same series, you just focus, and then you already, when it's in focus, you've got the correct working distance. Okay, so that's not something that you have to measure out. Yeah? You can see them in flower petals very well, the ring-shaped structures, Xylem. Yes, I spent like 2 months researching different microscopes and companies, so don't be afraid to do some work before you make a purchase. Yeah. There's a general recommendation that I have, which I've been mentioning over and over again. Sometimes I think research is important, okay, because you learn a lot during the research process. However, I think maybe sometimes it's better also to simply maybe buy a low-cost microscope and then just experiment around with it. Okay, that's basically the stomach of a dog. Okay, so that's a different approach really. You do a little bit of research. You say, oh, you set a budget. You say, okay, I'm just going to buy this microscope now, and then you work with it, you enjoy it, and then when you learn more later on, you can always, it's a long-term hobby, right? This here, because this was the question. You see, that is a animal tissue, dog stomach, and I think it becomes very evident already here that, or just let me flip it out here, that the individual cells are much more difficult to see, if not impossible to see. Yeah? It's kind of it goes back a little bit to one of the questions that one of the viewers had. Yeah, you see, and I think that's also one of the reasons why the early microscopists, the early biologists who were putting things under the microscope, they were really fascinated by plants. They were really fascinated by the cell wall, because the cell wall was simply so visible, and so prominent. Yeah? Here, the individual cells, I mean, this is a stained one. I think the individual nuclei can be seen as those dots, but you can see that you cannot really see where does one cell stop and the next one start, because animal cells don't have a cell wall, which is so prominent. Yeah. So this is a little bit the thing here. So, okay, that's a question to one of the viewers. Which microscope did you choose in the end? Okay. Are you going to look at commercial brand slides or water samples today? Well, actually both. This one is a commercial one, and I just showed you some water samples. I mean, I've got some water samples over there in the window. Yeah. Is there a proper setting for the height of the condenser and the size of the aperture? Yes. Okay. Okay, this is an interesting one here. So Bob asked the correct setting for the condenser. Generally, what you do is you have to move the condenser all the way up to the top. Okay, that is generally the correct setting. And the question is now, what is the correct aperture setting for the condenser? And I'm going to just show this to you now I'm going to now open and close the aperture here. Okay, and what you're going to see is that the image becomes brighter and darker. My camera adjusts the exposure time a little bit. Okay, so that means even though it's brighter now, the camera adjusts here. And what you're going to see, that when I open the condenser, you're going to see that the contrast goes down. And when you close the condenser all the way, the contrast goes up. But if you close it too much, what will happen is you're going to see diffraction patterns. So strictly speaking, what you have to do, let me switch back here again. Strictly speaking, what you have to do is for the best resolution, you have to set the condenser to a value at least the numerical aperture that is printed on here. So this means that if you're setting the condenser value, if you're opening it all the way up, okay, then you have a value which is higher than 0.65, and then you get maximum resolution. However, the image will also look a little bit low in contrast. Okay, and when you close the condenser all the way, then you get a higher contrast image, but at the same time you're losing resolution. So it is a trade-off, and that's why we have a condenser. But if you were asking for the single correct setting, if I'm supposed to answer that question, I would say set the condenser to the same value as the numerical aperture which is printed on the objective. Okay, this is the short answer. However, what I do in practice is I open and close it all the time until I get a picture which is sufficiently pleasing and yeah, clear. Okay. When would I move the condenser down? You would move the well, okay, I can also give you an illustration here. Let me do the following. Let me switch to a lower magnification now. So what I have is the following. You're seeing now a white spot here. I have so-called Köhler illumination, and better microscopes have this. And Köhler illumination improves the contrast. And what I've done now, I've closed. This is not the condenser diaphragm, but it is the field diaphragm. And when I move the condenser down and up, then you see that I'm able to focus the field diaphragm. And the correct setting generally is the time when I see the sharp edges of the field diaphragm. Okay, so the condenser is now not quite all the way to the top, but a little bit down. Okay, and then I open the field diaphragm. Okay, and this now is theoretically the correct setting of the condenser. And if you look over here, okay. Yeah, just beneath the slide, this is how I raise and lower the condenser. Okay. Yeah. Generally, there is the saying you keep it all the way to the top. Okay. But in my case, when I move it all the way to the top, then the field diaphragm is not in focus, and I have to move it down a little bit. Okay, until it's in focus. Yeah, but this does not change the focus. This does not change the focus of the image. Okay. And then you open it just enough, and then I also do not have a lot of stray light. So you are correct. Emotional attachment does play a role. I have been captured by a specific brand and being sucked into a deep black hole to become a loyal customer. Yes, yeah, that's why not? Okay, I'll tell you a story. When I bought my first microscope over 25 years ago, right when I finished my university studies, I decided, okay, I'm going to spend a little bit of money and I'm going to buy myself a microscope. And then I actually called up the company and I made an appointment with the guy there. At that time, Amazon did not exist, of course. There were also not as many microscopes available. So I had to go to one of the big 4 brands. Well, it happened to be Olympus at that time. No specific. Yeah, well, actually, the only reason why I chose that model is because there was a local office there. So I went there. I spent quite a bit of money, and I bought myself a microscope. And the guy who sold me the microscope actually said it is we want to support amateur hobbyists because ultimately we're interested in long-term relationships. Okay, this is basically the good news. Okay, that they said okay. We're interested in this long-term emotional tie, and I said sure, why not? And then all of a sudden, I took not every company sees it like that. I just want to show you another quote now, another question that I just also want to share with you. Hello. I have contacted a brand name to buy a microscope for 4,500 euros. So I took out the brand name and everything. It's not really important. I gave my consent for this quote. So somebody wants to spend 4,500, and I never got recontacted for me for payment despite my reminders. They're not interested in amateur microscopists. Very disappointing. Okay, so yeah, I would say sometimes it maybe depends a little bit on the person that you're dealing with and not the company itself, but of course things like this are a disappointment. Okay, and I've heard this from people mailing me from different companies. Okay, that they don't seem to be interested in selling me a microscope. Yeah? So this is a pity, of course. Yeah? I wouldn't say it's always the general rule, but at least there have been some within the mostly positive experiences, of course, also some negative experiences. Yeah? And this can, of course, also contribute a little bit to the emotional tie that one might have to a company or to a specific brand. But then I would say it's not the brand. Don't give up. Okay, it could be that, I mean, I always say, unfortunately, we're all human beings and not robots. Yeah, life would be much easier if we were all computers or ants. But because we're all human beings, also the salespeople are humans, and then maybe, I don't know, maybe he or she was ill or whatever, right? So this can be possible. So don't automatically assume that if you do not get an immediate response that they're not interested. Okay. Also try to use a school's friends or science expo's microscope if you can before you choose. I bought a great telescope after going many star parties. Yes, so that's one of the things. Being able to try out certain things is, of course, something nice as well. Can you use a compound microscope, maybe with a special objective, for some basic functions that you would normally use a stereo microscope for? Yes, and I have done this already. So what you do is the following. I don't have a flashlight. You use the low power objective. You put some object on here, okay, that you want to look at, and then you look at it with a strong flashlight from top. So you can do that. If you watch one of my spiders, this video last week or so, I made a video about a spider in my other channel. I actually used that method. I put the spider directly on here with light coming from the top. However, and that's the important thing, if you want to look at some PCBs, which are basically the print boards for electronics, it's not convenient, okay? And 4 times magnification here is already a lot. So while theoretically it is possible, it's not you're not going to be happy. Okay, and you also do not get a 3-dimensional view. Yeah? Will be nice to see your microscopy lab. As a matter of fact, in a previous video, I actually did make show you some pictures of my microscopy lab. It's a room with a table and my microscope, a huge computer screen, some light, a microphone here which you cannot see, whole bunch of cameras, and that's it. Yeah, it's really nothing big. And then I've got a second one in my cellar. That's where I'm basically also, it's a little like my microscopy studio. Do you have any book recommendations for pond water microbe identification? I know quite a few, but almost every time I see something, a good reference book would be handy. Okay, I've got the old edition here. It's in German. You can get it anyway, because there are beautiful pictures in here, which I'm not allowed to show you because of copyright reasons. I'm really worried about that. But if you go online and if you basically find this on Amazon, the new edition has a different color, is white. It doesn't matter that it's in German because it's mostly drawings anyway. And when you know the name of the organism, then you can Google for it. So this is a high recommendation. Beautiful drawings. I've so far found pretty much almost every water organism that there is, I've found in here. Okay, so this is really a recommended book. Okay. Okay. The Motiga almost got an Accuscope, but has the, okay, yes. Lens, surfing, nice micro, okay. If the condenser should be at the highest position, why is there a control knob that adjusts the condenser height? Because you can essentially remove the condenser. So what you do is you can actually turn it down so that the condenser can be detached. And indeed, I have also seen microscopes where the condenser cannot be lowered. So it was actually also good microscopes where they completely fixed it with screws. Okay. You should get as much working distance specification as possible. Problem is that cost increases exponentially. Okay. Yes. Hello, I have a $100,000 microscope. Is it bad? $100,000 microscope. Well, yeah, these microscopes do exist. Is it bad? Well, honestly, yes, if you never use it. The best microscope is the microscope that gives you the most satisfaction using. And if you're happy with it, and do not forget that scientists in the past, they did not have microscopes of the current quality, and they made important discoveries. Yeah, so yeah, thank you. And so Okay, yeah, so they're talking about Elon Musk now, okay, yeah. There is also this same book in English and Italian at least. Oh, really? I did not know that they have translated this, okay. That's good, okay, good to know. So let's go on here. What else is there? Wow, okay. There's some of the questions are quite long. Yeah, here. Love your channel. Thank you for all your generous contribution and time in making the videos. I'd like to suggest a topic for a video if you haven't already done it. A video on how to clean slides. I have already made videos, but I still want to show you some dirty slides under the microscope. Okay, how to clean slides and what to expect from new slides from a box. We just got a very nice stereo for the kids, but the biggest problem I've encountered is that the slides and cover glasses are included with the scope are absolutely horribly corrupt right out of the box. Handled with much care. There are many different types of corruptions, flaws, bubbles in the glass, foreign matter in the glass, scratches. Honestly, I have a difficult time imagining that there are bubbles in the glass. Could this be plastic slides? It's almost as if they included recycled, poorly cleaned slides. I won't mention the company because I love the scope, but it would be very valuable to see how to clean slides. Well, okay, I've got a slide box here, fresh slides. And I don't know if you're able to see this. I'm going to hold it against the light here. But they are really dirty. It's almost like they're, I don't know, I need a dark background. Maybe. Yeah. It's almost like there's a layer, almost a white layer on it. And I had problems washing this off with water. And water and soap wouldn't work. So what I had to do is I think this was some kind of a layer of calcium carbonate, maybe because they actually did wash the slides and they did not properly dry it. And maybe there's some kind of a calcium carbonate deposit on here. And if this happens, when it's not completely clean, then put it on, wash it a little bit in vinegar because the acid is going to remove the calcium carbonate. Okay, but you know what I want to do is I want to show this to you simply, a dirty slide out of the box. So let's, because this is almost, I think, almost not usable. Okay, here we go again. Okay, let me remove those two slides here. Let's put this in. Okay, let's go up with the magnification a little bit. Okay, let's focus. So this is how the slide looks like, a fresh slide out of the box. I mean, it is unbelievable. Okay. This is really unbelievable. And honestly, yeah, not usable. And for this reason, yeah, I have to clean the slides first, and that's a little bit of work. I've also tried to clean slides by putting them into a dishwasher. This also works, and because the dishwashing detergent also removes quite a bit of dirt. But this is a fresh slide, okay? It even says pre-cleaned and ready to be used. I cannot believe this, okay? Yeah, just saying that dirty slides are nothing, unfortunately nothing unusual, okay? Thanks, I think I may have to order that book. Okay, what is the title and the publisher of the book you recommend? Yeah, the title, and you can Google it directly, it's the German version, but maybe you, there apparently are English translation. Leben im Wassertropfen, Life in a Droplet of Water. Now I know that this title is also the title of other books, okay, but you have to go by the author, Heinz Streble, Dieter Krauter. Okay, Streble and Krauter, and it's a Life in a Drop of Water, and that is basically an identification book. Yeah, so Life in a Droplet of Water, you got to be careful because there are other books as well that have the same title. Yeah, Michael Fowler, founder of Freshwater, an identification book. Yeah. The green version here is an old version. Okay, they have now new versions as well. Yeah, it contains thousands of, I don't know, a lot of drawings. Yeah, and really nice. You might be able to have a short look in Amazon. Maybe they have a preview of the book. I've once seen a preview, but it did not include the pictures. Yeah, unfortunately. Yeah. I would trust the target grade before I would trust, okay, Alan, yeah. It is unfortunate to see an unresponsive representative. Some of the reps aren't interested in dealing with individuals. Some of them are nice enough. The third-party company might be able to help in this case. Yep. There are some companies actually that are reselling the microscopes. Okay, so maybe some intermediate retailers and so on. The book apparently has also been translated into Spanish. Okay. Okay. An atlas of microorganisms. So, okay, yeah. So what else do I have here? So that is a little bit the whole thing with the cleaning of slides. Okay. What else do I have here? What else do I have here? Okay. So this is something that says, heavens, please not. This one. How can I get the best focus with only an objective with 100 times? So apparently this person who wrote that has a microscope only with 100 times objective, probably 100 times oil immersion objective. And I say, no, forget it. You need to also have the lower magnifications, because you need to focus the specimen first with the lower magnifications before you can increase the magnification. Jumping directly to 100 times, I think is not gonna work. Okay. So, what else do I have? So that's another thing a little bit that can be discussed. Here, there's a complaint about unrealistically high magnifications. The Amazon link you provided for both of these microscopes go up to 2,500 times power. Are they so cheap that they need to market the high power to sell? I would think to stay away from a brand that markets this way. You always recommend not going for more than 1,000 times. Well, okay. Yeah. The point is the following. There is a certain resolution limit in microscopes, and this means that you cannot meaningfully magnify much more than around 1,000, 1,200 times. Of course, you can magnify higher. It's going to become more blurry. Now, there are some companies that advertise their microscopes with a 2,500 times power. And what they do is the following, is they will include, do I have some of these here? Yeah, they will include not only a 10 times eyepiece, but also a 25 times eyepiece. And if you use now a 100 times oil immersion objective and a 25 times eyepiece, you get 2,500 times. So basically, you're significantly increasing the magnification just by adding a very, very cheap eyepiece. Okay. So in other words, what I could also do is I could take a 40 times objective and a 25 times eyepiece, and I'm also getting 1,000 times. Or I can use a 100 times objective and a 10 times eyepiece, I'm also getting 1,000 times. But the thing is that by using this combination, 25 and 40, you don't get the same image quality as if you use a 100 times oil immersion objective and a 10 times eyepiece. So the resolution image quality is significantly determined by the objective. And by changing the eyepiece magnification, you can have any magnification that you want at the end. Just go up with that. But it's going to be more blurry. So those companies, I mean, they are selling, they're not lying, because the microscope does magnify 2,500 times, right? And it's nice to have such an eyepiece here with 25 times. I've never used it, but I mean, why not? Yeah. But the point is I question the value of this, right? As a matter of fact, if you buy microscopes from big brands, big four, for example, they don't even tell you in the specification the total magnification. Yeah. They just tell you the objective magnification. They don't even say, oh, the microscope magnifies up to, I don't know, 2,000 times. Yeah. Basically also the more expensive microscope brands, they do not even provide a 25 times eyepiece. And so, yeah, but the 10 times is the maximum that they have. So in that sense, it is indeed a little bit of a marketing thing, but still, yeah, it does magnify 2,500 times, but I do not consider it very meaningful. Yeah? Because already 1,000 times is difficult. I mean, the objects are the microbes. They're moving out of focus very quickly, and they're difficult to chase and so on. So it's not quite. It's very dark, the image. So I do not consider it very useful. But I mean, if you want to play around with it, why not? So that's a little bit the thing. I personally would prefer if we would go away from the power, because I'm receiving also emails and comments where people are asking me, well, is 2,500 times enough magnification? Is this good enough? Or do I need 3,000 times? I said, no, come on. That's, yeah. But we see a similar thing also in astronomy. I mean, I used to do a lot of amateur astronomy back in the day. And when they sold telescopes, the same thing. They went up with a magnification 500 times, 600, 650 times, 700 times magnification. But the real maximum limit, meaningful limit, is 300 times. So you see the same trend a little bit. It's not only in microscopy. To Elon Musk, would you be kind enough to donate the 100,000 US? Oh, that would be nice. I was already hoping that the real Elon Musk is watching me. So then let's move on here. Another interesting one about specimen mounting. Okay, I'm worried about the quality of specimen after mounting. I have specimens of stomata guard cells. Does cheap mounting media affect the specimen quality? Thank you. So very briefly again, and we can look at those as well. When you make those permanent mounts here, then the question is what about the quality of the mounting medium that you use? Does the quality of the mounting medium determine the quality of the specimen durability? And I say, well, it's complicated. It is not really the quality as such that is important, but it depends a lot on how well has the specimen be prepared. It must be complete. For example, for some specimens, they must be completely dehydrated and dry. That's very important. If it's not completely dehydrated, then the specimen, is going to start to decompose maybe. Right, and then you have bubbles, and it depends on a variety of factors, and not necessarily alone about the price or the cost of the mounting medium. So there are other factors that are involved. Generally, if you get yourself a mounting medium, then you have to choose the mounting medium depending on the specimen that you want to mount, because different mounting media are better suited for certain specimens, and then you have to also prepare it. And I would say that these are the things that determine ultimately the quality and the durability of the mount, and not the mounting medium itself. Okay, so I mean, it's Hydra. Let's see. Because let's see if I'm able to find it. Hydra is a water organism. I don't know if have I even looked at this one before. It's completely out of focus as well. That's not Hydra. You see? Here it is. Here it is. I think it is. Here it is. Okay. It seems to be also a cross section. You see, even this commercial slide is kind of dirty in the background. I have one of those 2,500 times microscopes, and it's a waste of time. When it's trinocular, you can't capture it anyway through the photo tube. To be honest, the quality with a 60 times objective is poor too. Let's put it this way. The higher you go up with the magnification, the more blurry the image will become. I mean, if you use, for example, 40 times or 60 times objective, then the value, the numerical aperture, this value over here is going to be higher, and the resolution is going to be higher. Okay, so actually you're going to see more details. However, the magnification increases faster than the resolution. So this means that subjectively, the higher you go up with the magnification, subjectively, the more blurry the image will become. And also because of the lower depth of field, the image might appear to be blurrier, but objectively, you're going to see more details. I know this sounds contradictory. Okay, from the psychology, you think the image might be more blurry. We're using a 60 times compared to a, let's say, 10 times. But actually you are seeing more details. So there is this difference between subjective perception and the actual details that you see. Okay, I tried the Elmer clear glue you showed in the video and it worked great. Yes. Oh, that's nice. For those of you who don't know, because many mounting media are difficult to get, are solvent based. You have to dehydrate the specimen starts to shrink. Some solvents are not healthy. I've basically recommended the so-called Elmer's clear glue, which is a PVA based glue, and PVA indeed is used as a mounting medium also for microscopy. So it's not even that far-fetched. I therefore, ever since I discovered Elmer's clear glue, I recommend it as a general purpose mounting medium, which is also safe to use, and it's water-based, so you do not have to completely dehydrate the specimen, and it seems to work, and it also doesn't shrink so much, so I find it as a very practical solution. Okay. Hi, you use a lot of Uperol, that's correct, but I couldn't find it in the internet available. Is there any other brand similar with the same properties? Uperol is actually not a brand as such, but it is actually the name of the mounting medium. Uperol is very commonly used by people who are mounting insects, and it is still the choice. And I know that it might be difficult to get, and I know it is possible to get it but you need to contact a chemical supplies company. Those companies that supply labs, and then it's a question of whether they're able to supply it to individuals. I got the Uperol. I got it from a shop that was also selling tools, equipment for people who are collecting insects. Okay, so but I admit it's rare. Okay, Uperol is liked by people who collect insects and who mount insects because it has a clearing activity. This means you put the insect into Uperol, and after a day or two, it becomes more transparent. Okay. Checking the relative products with Big Four for such claimed products as Amazon could be good approach. If the Big Four even don't have it, it tells us everything we need to know. Okay, I get it. So basically, because the Big Four companies, if you go online, okay, and if you check, then they will never advertise their microscopes with the maximum total magnification. Okay, because that's considered irrelevant. Yeah? They will only, for example, for my microscope that I have here, there is not even a 20 times or 25 times eyepiece, I think. Yeah? They only have 10 times. And the only choices that I have between the 10 times is the field of view, how much am I able to see. So you see that they place a focus on completely different things. Yeah? If you were ordering supplies and just setting up a microscope area for the first time, what would be the main thing you would order? Obviously slides, but different types of slides, pipettes, anything else. So what I would order, so basically if I'm were to start hobby microscopy, and if I'm setting up everything, what would I order? Well, I would definitely, of course, you want to have some microscope slides and cover glasses. It's easy, yeah, to get. I would order myself any kind of slide box. Okay, I almost say any kind because you just want to look at things, different things. Also, you want to show some of these slides to other people, you know, kids, whatever, friends, family, who quickly want to see something. Then what I also found useful are those small Petri dishes for water. Okay, for keeping water. That's quite useful. Yeah. I also have a whole bunch of jars standing around with water samples. Yeah, and what I really like is because I do not have running water. Yeah, so a water bottle like this. For cleaning, I've got another one with alcohol. Yeah, I have very important. I consider scissors like this. They're very useful, and the most important tool maybe some tweezers, so you can pick up some samples. But that's pretty much it. If you want to be cool, a pipette. This was actually, this is a microliter pipette, actually one of the cheaper ones where you can actually adjust the volume here. So that's a milliliter maximum. Yeah, and if you push the button up here on top, can you see this? Okay, you can, yeah, it pushes off the tip. Yeah, so it's it's a little bit of a luxury, but I like using it. Yeah, so they are basically, depends what you want to do, but ultimately, you don't need a lot. Yeah? I've been trying to find another hydra for years. Very elusive organism. Volvox seems to be less common as well. I found the hydras in pond waters. You have to collect some sediment, some leaves. Yeah. Motic is on my wish list. A very nice image quality. Yep. Okay, so let's move on. What time is it? What time? Oh, it's 1 hour 13 already. Okay. Yeah. So again, another interesting one. A little bit similar to some of the questions that I got here. I'm a total beginner and I want to buy a certain microscope. Please make a review of it. Okay. I can't, because I don't have the microscope. So that's a little bit the thing. However, and the reason why I'm showing this to you is if you watch the channel here, and many microscopes are very similar. Okay, so if it's an introductory microscope, then you just watch a review video of another introductory microscope, even if it's of a different brand, because this way you still get a little bit the information so that you can compare the microscopes better. Yeah? It doesn't have to be exactly the same, and as soon as a certain model is out, then they keep on changing it every couple of years. Yeah? So my suggestion is that even if there is not a review of exactly the same microscope, many microscopes are similar. Okay. So this here seems to be a little bit of a misunderstanding here. The resolution parameter was not explained in one of my videos. Is 0.65 higher or lower resolution than 0.17? Yeah, it's higher, because 0.65 is more than 0.17, but the 0.17 here actually is the thickness of the cover glass. Okay, so it actually means something different. So I think you're kind of confusing or mixing the numerical aperture, which is the resolution of the objective, with the cover glass thickness, 0.17. Okay, so this is, I think, something that I simply wanted to clarify here. And the 160 refers to the standard which is used, and of course the 40 is the magnification. Okay. I bought a dissection kit like I used to have in zoology lab, including clamps, forceps, scalpels, and other cool stuff. Yep. Yes, if you go online on Amazon, there are a lot of dissection kits available. I got a sample from a river 2 days ago and it smells like sulfur. Let me guess. You wanted to say it smells like sulfur. And the reason why your water sample probably started to smell bad is, let me guess, probably because you put a lid on top. Excuse me. Okay. So look at this here. Water samples, when you collect water samples, you can put a lid on top, but make sure that there is a lot of air in here. Okay, then you can put a lid on top. But if you fill up the water sample all the way to the top and you put a lid on top, then it's going to grow anaerobic, and sulfur, hydrogen sulfide is going to be produced. Their decomposition starts. Okay, and that's not good. And then basically oxygen level drops and many of the microorganisms will die off, and some bacteria will start to reproduce. But the interesting things will die off. So my suggestion is, if you want to keep a water sample and if you really want to put a lid on top to prevent it from spilling, make sure that there's sufficient air in here, or keep it open. Yeah, I think putting a cap on top with a lot of water, after a few days, it's going to start smelling bad. Okay, decomposition will start. If you are in the US, check your local Goodwill for glassware. Sometimes you will be surprised what you can find at very low price. I'd found a couple of useful glasswares and reference books. Do you have any videos on Vorticella? Yes, of course. In my other channel, I just a couple of weeks ago made one. I found some recently in one of my live streams, and there were a lot of fun, constantly impeding the traffic of other ciliates. And yes, I did make some Vorticella video as well. Okay, so let's move on. Yep. So this one is now referring to cameras. It's a long question, but I actually it's not so difficult to answer. Thank you so much for providing us with all your knowledge. It has helped me so much with learning to understand how microscopes work. I recently brought the Bresser Biolux. Okay, I additionally added the Bresser Full HD Microscope USB camera because I want to create high quality and sharp video and images. Unfortunately, I noticed the Full HD USB camera creates more grainy and noisy images than the HD camera. And this does not surprise me. Okay, now I think I have a theory of why this is the case, which I would like to explain. I would love to create the images that are sharp. Okay, could I create the same type of images if I attach the photo camera through an adapter to the microscope, or would you buy a higher quality one? So, the point is the following: you buy a new camera with a higher resolution and the picture looks more grainy. How is this possible? One reason could be that the sensor size, if you try to pack more pixels on the small sensor, then the image will become grainy. Okay, so you see, there are multiple factors that determine the image quality, and what I would suggest is you go down with you go into the software, you go down with the gain of the camera, and you increase the light intensity. This will also reduce the grain significantly. Okay, but the problem here is not a problem. It's not the problem with the camera. It's simply in the law of physics. If you have a lot of pixels in a small area, then the image will become more noisy and more grainy. That's the case with any camera. That's simply the way that digital cameras work. Okay, so I know people expect a better image quality because they go up with the resolution, but actually, yeah, you have a higher resolution, but you're losing quality somewhere else. Yeah, but you can compensate this, go down with the gain in the software, increase the light intensity, and this way you can actually tweak out less noise. Okay. Okay. If I open it now, it will smell in the room. Yeah, so basically my suggestion is if it starts, the water sample starts smelling bad, then I would simply throw it away. Okay, because then decomposition has started and doesn't smell so nice. Okay, you know what? Because I've been showing you the desktop the whole time, maybe I just want to show you some other things that are quite nice. Yeah, so I don't know. Maybe I've shown the sunflower is usually also quite nice to look at, so that we also look at a few. Yeah, that was the Hydra from before. Okay, dirty slide. And here we have the sunflower. That's why I'm saying plant cross sections. That's why get yourself a slide box. So see, a sunflower is a so-called a dicot plant, and therefore all of the vascular bundles are arranged on the side here. Yeah? So again, cell walls easy to see, and the individual cells can be seen. Yeah? Typical for plant cells. Okay. It has a C-mount screw thread. Okay, well, just a second. The Raspberry Pi camera makes a good cheapish alternative to the camera people sell for microscopes, although lately getting Raspberry Pi is a challenge. Yeah, I've also heard about that people actually connect the camera of a Raspberry Pi. What you can also try to do, if you want to experiment around a little bit, is you can get yourself a webcam, and you can try to disconnect because the webcam is a wide angle, right? But you can try to do the following: you can try to remove the optics of the webcam and directly connect the webcam then to the microscope. Okay, so without the eyepiece, however, so that the objective projects the image directly into the webcam. This was actually my very first digital camera. Was actually an old 640 by 480 webcam that I directly connected to my trinocular port of my microscope. Yeah. It actually worked, but the resolution was very low. But yeah, it kind of worked. Okay. The Hydra tentacles aren't long like the Hydras like in your videos. The tentacles are much smaller and shorter. Is it because of the mounting media? Should not be because of the mounting media. Sometimes the Hydra will actually contract, and it could be that maybe during the preparation, they contracted, or during, I don't know, this kind of looks a little bit like it has been microtomed. This does not look like a whole mount. Go up a little bit here. Look how thin this is. I honestly, I think that this is actually not the complete Hydra, but actually a cross section, and it could be that therefore part of the Hydra was actually cut away. Yeah, it is. Of course, it's a cross section. It must be. I will tell you why. Let's do a little bit of biology now. Okay, so let's go all the way down with the. Okay, look at this, how dirty the slide is. Okay, so let's. I'm going to explain you now a little bit why this is a cross section. What you have. Let's look at the thing here. The tentacles are here. They should be much longer, obviously. This is the foot of the Hydra, and look, this is the stomach on the inside. So the food is actually drawn in, is digested in here, and the rest is spit out again. Yeah, and the reason why the tentacles here they are so small is because, well, they were not properly prepared. So I would not blame the mounting medium as such. I would rather blame the fact that this Hydra has been microtomed and has been sliced, and probably during the preparation process they kind of removed the tentacles. But look, look at all of those dots here. This slide has not been cleaned. Okay, and this was, yeah, that's what I call a fairly low quality preparation. Okay. What I would try soon is to connect my smartphone to a trinocular with a C-mount extension. Ah, that's interesting. Okay, connecting somehow a C-mount, yeah, looks like a body cross section, and maybe a 3D printed adapter for the iPhone. Yes, I made also recently a review of one of those iPhone adapters. I see a double image on my compound microscope. What might be going on with it? Okay, for double image, you do the following. Two things that you have to adjust. Number one, you have to adjust the distance of the two eyepieces, number one. And number two, it could be that there is almost certainly a diopter adjustment. There is around the eyepiece a ring to turn. And if you have unequal eyes, like one of your eyes is worse than the other, then the two eyepieces have to be adjusted that each eye is corrected. Otherwise, one eye will be in focus and the other eye will not be in focus, and then your brain will have a problem in combining the images. So adjust, if you see a double image, adjust the distance and make sure that the diopter adjustment is correct. Okay. You can get them with some microscopes. Mine came with. Why is the Hydra a bit purple? What stain do you think? I don't know the stain that they used, but and that's a little bit one of the criticisms of some samples, is it that there is not enough information. It just says, yeah, Hydra LS. LS stands for longitudinal section, but it does not say anything about the stains used. Okay, that's a little bit of, indeed, a little bit of a, yeah, of a pity. Yeah. If it looks a little bit, I don't know, here now it looks a little bit bluish, purplish, could be methylene blue, I don't know. Yeah. Okay, thanks. That makes yeah, makes sense. I recently had to get glasses for my eyes. If you have glasses for your eyes, then you have to set both diopter adjustments to the same, because the glasses will already correct it. But be careful, it's another thing, I'm talking from experience here, because if you use eyepieces and if you're wearing glasses and you're always touching the glasses, make sure that your glasses don't become scratched or the eyepieces don't become scratched. Okay, just talking from experience here. Okay. Would that be a good idea to grow Hydra with the use of a once they are cut in pieces, they will start to repair and grow? Not with a blender. Okay, don't do a blender. The reason is the following: if you use a high-speed blender, you might actually chop apart the individual cells. Okay, because blenders are used to actually chop apart cells so that the DNA is released. Yeah, so that would be a little bit too much. Okay, so if yeah, and generally Hydra, if you feed them properly, they will reproduce on their own. And if you chop them apart and if there is not the right conditions present, then they will also not properly regenerate anyway. Okay, yeah, so I talked to a colleague of mine who's got an aquarium, and he said Hydra in some aquariums are a huge problem because they reproduce so quickly that, yeah, that's actually difficult to manage them. What's the difference between a cross section and a longitudinal section? Basically, when you have a cross, let's put it this way, I don't know, some object. Yeah, I'm gonna use I'm gonna use this here as an example. Okay, cross section would be if I cut it like this, okay, and look at it from the side. Yeah, and longitudinal section would be if I cut it lengthwise. Yeah, so that's essentially simply the direction and the way it's cut. Okay. The Amscope or Swift USB 3 cams are money well spent. The included software is quite good for complete control compared to free apps. Mild soft zoom can be quicker than changing objectives too. Yep. So yeah, USB 3 cameras, and if you have a sufficiently fast computer, you can also capture sufficiently fast video with that. Sometimes USB 2 cameras are a little bit slow for videos, but USB 3 cameras, provided that your computer is fast enough, is also able to capture video, which is nice. Okay, I just got my first microscope. Could you help me figure out how to use the methylene blue powder that came with my kit to see cells? What fluid do I mix it with? That's an interesting one. And what concentration? Now, you can mix it, at least the Löffler's solution that I have over here is a mix containing water and some alcohol. My suggestion is that you mix the methylene blue with a little bit of distilled water first. Okay, but mixing the powder with water, it might not always dissolve. Okay, so that's essentially some of the things. Yeah, I think the reason why they included powders is because they're not allowed to ship fluids anymore. So I've had difficulties actually getting liquid stains because it's not allowed anymore to, or not easily allowed anymore to mail fluids and liquids, and that's why sometimes they will include powders. Okay, but there are different types of methylene blue recipes, and I would suggest that maybe you Google a little bit on the amount that you have to mix it. Okay, will deionized water work as a substitute? Well, deionized water is distilled water. Okay, so that should not be a problem. Yeah, so yeah, let me see something else. So this was the Hydra part. Okay, and some questions. I already forgot what I answered. Okay, this was the providing with us all the knowledge for full HD. Yeah, I think I'm pretty much through with all of the questions that I've got. Unless you have more questions. I mean, it's already 1.5 hours anyway. It's quite a long. I know that some of you were really desperately waiting for some water samples. Honestly, I have the following. I'm going to try now the following. And I think it might not, I don't know. I have a little mini aquarium here. Look at this. So that's actually something I recommend as well. Yeah, it's a huge jar. Okay, and in this jar I have lots of green stuff floating around. This is already, I don't know, over a year old. Water will always evaporate, and therefore I always keep on adding a little bit more water to compensate for the evaporation. And honestly, I gotta be honest with you, I don't know what I can expect. Yeah, some of the algae might not be so totally interesting that we have here. So what I'm going to do now is I have to, wow. I just realized that I only have the dirty microscope slides left over. Yeah, so I have to clean this one here first. I'm gonna dry wipe it, and it's not gonna be totally clean. Better than nothing. So, okay, as a last little try today, let's do a little bit of water microscopy. So I'm going to pull out, and sometimes indeed there are some interesting things to be seen, sometimes not, and sometimes there is stuff in the sediment. Sediment basically means all the way down here, right? Yeah, what I'm going to do now is the following. I'm going to go in here now, pull out a little bit of more water. Okay, then I need a cover glass. I'm super organized today. Where did I put my cover glasses? Okay, I'm gonna use, I've actually wanted to use the large cover glasses. I put them somewhere. Don't know where they are, so I'm just gonna use the small one here. Okay, and let's see what we're able to find. just because some people want to see a few living things, and then usually there's always this demonstration effect that there's nothing of interest. That's a little bit my worry sometimes. Yeah, at least some algae. Okay, what are we able to see here? It's a little too thick, I would say. Just a second, let's go like here. Yeah, whole bunch of filamentous algae. No water fleas so far, no paramecia so far. But some small water life. Not a very interesting sample. Some months ago, there were tons of mites in the sample. But there's one thing that I recommend you to do if you have a water sample like this. You just wait, and then all of a sudden you might be able to see the formation of air bubbles, or oxygen bubbles rather. But this would take too much time. But this is also something that I've done before. Look at those bubbles. If you monitor them over the course of several minutes, half an hour, you're going to actually see that they will start to grow, because this is photosynthesis happening. Okay, so let's here now at the higher magnification we're a little bit better off. What is this? Those dots are too big for bacteria, I would say. Yeah. But maybe if we just watch this for a couple of minutes, maybe we're able to see that. Yeah, let me turn up the light a little bit more for more light. Maybe we're able to see that hopefully that those bubbles will grow, because this would be then oxygen which is made by the algae. Okay. So when I got the sample, there were 10 to 12 huge worm-like creatures in the algae. What are those worm-like creatures? Either they were really worms, nematode worms, or more likely they were, if they were kind of inchworming along, most likely some rotifers. Okay, or yeah, some segmented worms. Yeah, nematode worms could be. Yeah, flatworms could be many things. Do you refill evaporated water with regular water or distilled water? With regular water. Please don't use distilled water, okay? For osmotic reasons. And if you you be careful, because when you use tap water, make sure it's not chlorinated. It depends a little bit where you're living, or you use non-carbonated mineral water, just yeah, still water. But you see that the bubbles are already growing a little bit. Yeah, that's oxygen being produced. Yeah. So in that sense, you see that there is quite a bit of biological activity going on. Okay. Are you mean visible to the eye? I don't know what this refers to now. It said huge worm-like creatures. I wonder how if they're really huge, then you should be able to see them with your eye. Yeah. A bit weird question, but why is water, or at least some water, blue? Is it chemicals, and why wouldn't you see it under the microscope? Well, the thing is the following. Why is some water blue? Well, tap water, I mean, is not blue, it's clear. The blueness that you see in the ocean and in lakes and so on, that's sometimes the reflection of the sky. Obviously, or do you mean when there is a background like blue like this? Well, that is because of the filter system that I'm using. Okay. What is the difference between regular and distilled water? Distilled water is pure H2O, no minerals. Okay, just water. If you get water out of the tap, then this is of course water, but it contains also many minerals. At low concentrations, and so it's not pure in that sense. For chemical experiments and so on, you need you don't want those minerals. You want to have pure distilled water, but essentially the pure distilled water can be also a problem if you add too much of this to water life, because water life does not want distilled water. There are lots of minerals and salts and so on around it. Yeah. Exactly, the ions. Yeah. Hello from Greece. Yeah, but unfortunately, I'm slowly starting to stop now. What is the best stain for viewing live samples? Honestly, none. if you use a stain, chemical stains, and if you add it to water samples, then in many cases the stains will kill the organisms. So you have to be a little bit careful, and if you use any stains, you have to use it in very low concentrations. So my suggestion is methylene blue does work for live samples, but only at low concentrations. So that is the thing. No water organism. Oh, plenty of them. Teeny is saying no water organism. I actually see a lot of them. Look, the algae. Look. Up, where's the arrow? Just a second. Now the arrow is going the wrong way around. I mean, look at all those dots. These are all microorganisms here. Algae producing oxygen. Okay. Probably some bacteria in the background, but I think you were probably hoping for larger organisms, like rotifers and ciliates, large ciliates, and indeed they are not visible here. Okay, unfortunately, but there are plenty of organisms here, like for example, all of these algae that you see. Okay, probably some diatoms growing on here on the surface. Yeah, but I get it. I get it. You want to see a little bit of movement, a little bit more action. Okay. Then dots are micro. Yes, all of those dots are microorganisms. I don't know what they are, but I get it. All of those dots floating around here in individual cells. But you know what? Okay, fine. Let's remove this again, and let's look at a different, from a different location. Okay, let me see. I don't know if there's a lot of interesting stuff here. I don't know. Let me check. I'm a little bit suspicious of whether. There's going to be something interesting here. Okay. And let's put this one on top here. Again, another cover glass. So I took some water now a little bit from further down, and I need some tissue paper because there is too much water here. And I don't want any water spills over the edge of the microscope on the microscope stage. I don't know, no guarantee, but let's have a look. If we're a little bit more lucky here. You have plenty of algae, single-celled algae now. And if you look carefully, there is some movement going on in the background. But otherwise, I do admit that the organisms in this sample are not very large. Okay. Well, not every demonstration is always successful. Okay. Small leeches all over my ankles when walking through the rainforest. Yep, diatoms. Fluorescent protein. Yeah, that's kind of advanced. Okay, but you know what? If you really want to see something exciting, then I think I'm going to maybe for the next time prepare. Maybe I'm able to find some tardigrades or so in moss, and then I can show them to you as well. But yeah, we'd have to go up a little bit. Let's go up a little bit with the magnification. Maybe we're able to see a little bit more. And these are individual cells here. All algae, a lot. If it's green, it's an algae. And some diatoms as well. See, at this magnification, we're able to see a little bit more. Yep. That's actually also one of the, how do you say, exciting things about microscopy. Sometimes you don't know what to expect. There's always a little bit of a surprise. I remember that some time ago there were lots of rotifers in this sample, but there's a natural progression happening. Yeah. Okay. Yeah, diatoms. Those. Do you see those little tiny things here? These are diatom shells. Okay. Those oval regular structures that you see here are diatom shells. Okay, well, what I'm gonna do now, I think after 1 hour and 43 minutes is, I think I'm slowly going to call it quits for today again. But there are a couple of questions. So do other subjects such as biochemistry, immunology, molecular biology, and many others, okay? Yeah, shells. Yeah, shells, because diatoms are algae that have a silica shell, a hard silica shell on the outside, and when the cells die off, then the shell remains, is left over, and this is something that yeah, looks quite nice, and you can actually see them. Yeah, usually they sink down to the bottom of the glass jar or the pond, and when you take specimens from there, then you're able to see those shells. Yeah. So it's basically it's made of silica, yeah. And silica is like glass, yeah, the same similar chemical. And I have to tell you that those diatoms here are also kind of a little bit small, yeah. But I don't know, maybe I can readjust this a little bit. Okay, just a second. Maybe this is now it's a little bit better. Yeah. You see that all of those regular structures, these are all silica shells of diatoms. Okay, folks, I think it's time to call it quits for the day. I'm slowly also starting to feel a little bit tired. Okay, yeah, I would like to wish all of you the best, yeah, I'm always happy when there are comments in the chat. And for next week, I'll think again of something to put under the microscope. Yeah, it's wish you all the best. Happy microbe hunting as always. Okay, and see you around next time. If you have any questions, please remember them, and then you can post them again in next week's chat again. Okay, I'm really gonna call it quits now because I'm already quite tired. Okay, then all the best, and see you again. Bye bye.